Hello friends, welcome to this lecture on metabolism 1. We'll discuss about the carbohydrate metabolism in this lecture. Here are some previous year questions that has been asked in June 2015 on carbohydrate metabolism. The glycolysis and citric acid cycles are important pathways to generate energy in the cell. Even below are the statements regarding the production of ATP. And four statements are given, out of which we have to select the correct options. Then in June 2015, another question was asked regarding the pyruvate dehydrogenase. It is subject to feedback inhibition by its products in glycolysis. Some of the chemical compounds which might be involved in the process are listed below. So we have to identify which compounds they give the feedback inhibition to the pyruvate dehydrogenase complex. So let us first have a look at some of the basic aspects of the glycolysis and TCA cycle, and then we'll discuss about these questions. Glycolysis, it is derived from the Greek words, which means the splitting of sweet. During this process, one molecule of glucose is degraded into two molecules of pyruvate. Free energy which is released in this process is stored as two molecules of ATP and two molecules of NADH. So here the overall reaction of glucose is glycolysis is shown, where glucose is split to two molecules of pyruvate and two molecules of NADH is released. And this can result in further production of ATP upon oxidation of NADH via oxidative phosphorylation. So if we look at the overall free energy change of the glycolysis up to pyruvate, then it is about minus 85 kilojoules per mole of glucose. In standard conditions, glycolysis is an exergonic reaction which tends to be irreversible because of negative free energy change. So if we look at the fate of glucose in living systems, the glucose can be further oxidized to carbon dioxide, completely oxidized to carbon dioxide in presence of oxygen. And in case of anaerobic condition, in absence of the molecular oxygen, the glucose can be oxidized up to pyruvate level only. So free energy change in aerobic condition upon oxidation of glucose to carbon dioxide is very high, and in case of anaerobic condition, the free energy change is very less. So about 5.2% of total free energy that can be released by glucose is released in glycolysis. And the glucose, you can see that it can be subjected to storage in form of glycogen or starch or sucrose in different organisms, plants, and so on. Glucose can also be used for the synthesis of the intermediates like ribose 5-phosphate via pentose phosphate pathway, or it can be oxidized to pyruvate via glycolysis. Under anaerobic condition, the pyruvate that is formed can be subjected to ethanol production or lactic acid production. But under aerobic condition, pyruvate, which is formed upon glycolysis of glucose, can be then further oxidized to acetyl-CoA, which enters into the TCA cycle and gets completely oxidized to carbon dioxide. There are 10 enzymatic catalyzed reactions in glycolysis, which occurs in two stages. Stage 1, in which the reactions up to reaction 1 to 5, they are called as preparatory stage, in which glucose is phosphorylated, converted to fructose, which is again phosphorylated and cleaved into two molecules of glyceraldehyde triphosphate. In this phase, there is an investment of two molecules of ATP. And in the stage 2, which is called as payoff phase, the two molecules of glyceraldehyde triphosphate are converted to pyruvate, and concomitant generation of 4 ATP molecules and 2 molecules of NADH occurs. Thus, there is a net gain of 2 ATP molecules per molecule of glucose in glycolysis. Importance of phosphorylated intermediates. This is very important because the attachment of phosphate group imparts a negative charge which inhibit the diffusion of the metabolites through the membrane. It also serves for the conservation of free energy in high energy phosphate bond, and the attachment of phosphate group facilitates in the catalysis of the metabolites. Here is the first stage of the glycolysis called as preparatory phase, where glucose is first converted into glucose 6-phosphate by the enzyme hexokinase enzyme, where one molecule of ATP is used. and then glucose 6-phosphate is converted into phosphohexose fructose 6-phosphate by phosphohexose isomerase enzyme. Fructose 6-phosphate is then converted into fructose 1,6-bisphosphate by phosphofructokinase enzyme, which is one of the key enzymes of this pathway. Fructose 1,6-bisphosphate is then converted into glyceraldehyde 3-phosphate by aldolase enzyme, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So this splitting of fructose 1,6-bisphosphate is carried out by aldolase enzyme. And then in the payoff phase, the glyceraldehyde 3-phosphate, which is there, and dihydroxyacetone phosphate is then isomerized to glyceraldehyde 3-phosphate by triose phosphate isomerase enzyme. In the payoff phase, the two molecules of glyceraldehyde 3-phosphate are converted into two molecules of 1,3-bisphosphoglycerate by the enzyme glyceraldehyde 3-phosphate dehydrogenase enzyme, which results in generation of a reducing power per molecule of glyceraldehyde 3-phosphate. So from two molecules of glyceraldehyde 3-phosphate, two molecules of NADH are generated. Then from two molecules of 1,3-bisphosphoglycerate, 3-phosphoglycerate are formed by the action of phosphoglycerate kinase enzyme, which results in ATP synthesis. So here also two ATP are generated. The 3-phosphoglycerate molecule is then converted into 2-phosphoglycerate by the enzyme phosphoglycerate mutase enzyme. So here then the 2-phosphoglycerate is then converted into phosphoenol pyruvate by the enzyme enolase enzyme, and phosphoenol pyruvate is then converted into pyruvate. So two molecules of phosphoenol pyruvate are then converted into two molecules of pyruvate, which also results in synthesis of ATP at this step. So this is the second ATP forming reaction which occurs through substrate level phosphorylation. In both these cases, at these two steps, the substrate level phosphorylation occurs. Hexokinase reaction, which catalyzes the phosphorylation of hexoses. This enzyme is present in most cells. In liver, it is called as glucokinase enzyme, and it is the main hexokinase which prefers glucose as substrate. It requires Mg-ATP complex as substrate, and uncomplexed ATP is a potent competitive inhibitor of this enzyme. The enzyme catalyzes the reaction by proximity effect, bringing the two substrate in close proximity. This enzyme undergoes large conformational change upon binding with glucose. It is inhibited allosterically by glucose 6-phosphate, and it requires Mg+2 ions for its activity. Phosphoglucose isomerase or phosphohexose isomerase, which catalyzes the second reaction, it catalyzes the isomerization of glucose 6-phosphate to fructose 6-phosphate. This enzyme catalyzes the reversible isomerization of glucose 6-phosphate to fructose 6-phosphate, which is a ketohexose. This enzyme requires Mg+2 ions for its activity, and it is specific for glucose 6-phosphate and fructose 6-phosphate. The third enzyme of this pathway is phosphofructokinase 1, which catalyzes the transfer of phosphoryl group from ATP to C1 of fructose 6-phosphate to produce fructose 1,6-bisphosphate. This step is an important irreversible regulatory step, and the enzyme phosphofructokinase is one of the most complex regulatory enzymes with various allosteric inhibitors and activators. ATP is an allosteric inhibitor for this enzyme, and fructose 2,6-bisphosphate is an activator for this enzyme. ADP and AMP also activate phosphofructokinase 1, whereas citrate is an inhibitor for this enzyme. This enzyme requires Mg+2 ions for its activity. Aldolase reaction. This is the fourth reaction catalyzing enzyme, and it catalyzes the cleavage of fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This enzyme catalyzes the cleavage of fructose 1,6-bisphosphate by aldol condensation mechanism. As shown below, the standard free energy change is positive in forward direction, meaning it requires energy. Since this product of this reaction are depleted very fast in the cells, this reaction is driven in forward direction by the latter two reactions. Triose phosphate mutase reaction is the another reaction. Conversion, it catalyzes the conversion of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. This is a reversible reaction catalyzed by acid-base catalysis in which histidine 95 and glutamate 165 of the enzyme are involved. So this enzyme catalyzes the isomerization of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. And so from this reaction onwards, two molecules of glyceraldehyde 3-phosphate are made available, which are then acted upon by the further enzyme, glyceraldehyde 3-phosphate dehydrogenase enzyme. So this glyceraldehyde 3-phosphate dehydrogenase enzyme catalyzes the conversion of glyceraldehyde 3-phosphate to bisphosphoglycerate. This is the first reaction of energy yielding step. Oxidation of aldehyde drives the formation of a high energy acyl phosphate derivative. An inorganic phosphate is incorporated in this reaction without any expense of ATP. NAD plus is the cofactor in this reaction, which acts as an oxidizing agent. The free energy released in the oxidation reaction is used in the formation of acyl phosphate. So here glyceraldehyde 3-phosphate is phosphorylated to 1,3-bisphosphoglycerate and the free energy change is 6.3 kilojoules per mole. Phosphoglycerate kinase reaction is the next reaction which transfers the phosphoryl group from 1,3-bisphosphoglycerate to ADP, thereby generating ATP from ADP. So the name of this enzyme indicates its function for reverse reaction. It catalyzes the formation by proximity effect. ADP Mg complex bind on one domain and 1,3-bisphosphoglycerate binds on the other domain, and a conformational change brings them together, similar to hexokinase reaction. This reaction and the sixth step are coupled reaction, generating ATP from the energy released by oxidation of 3-phosphoglyceraldehyde. This step generates ATP by substrate level phosphorylation. So here the ATP is formed by the substrate level phosphorylation of ADP to ATP. Phosphoglycerate mutase reaction is the next step, which converts the 3-phosphoglycerate to 2-phosphoglycerate. In active form, the phosphoglycerate mutase is phosphorylated at histidine 179 residue. There is transfer of the phosphoryl group from enzyme to 3-phosphoglycerate, generating enzyme-bound 2,3-bisphosphoglycerate intermediate. This compound has been observed occasionally in reaction mixture. In the last step of reaction, the phosphoryl group from the C3 of the intermediate is transferred to the enzyme and 2-phosphoglycerate is released. In most cells, 2,3-bisphosphoglycerate is present in trace amount, but in erythrocytes, it is present in significant amount. There it regulates oxygen affinity to hemoglobin. Free energy change of this reaction is 4.4 kilojoules per mole. Enolase reaction is the next step where dehydration of 2-phosphoglycerate to phosphoenol pyruvate occurs, and dehydration of the 2-phosphoglycerate by this reaction increases the standard free energy change of hydrolysis of phosphoanhydride bond. The mechanism through which it works is rapid extraction of proton from C2 position by a general base of an enzyme, generating a carb anion. The abstracted proton is then readily exchanged with the solvent. The second rate limiting step involves elimination of hydroxyl group, generating phosphoenol pyruvate. The free energy change for this reaction is 7.5 kilojoules per mole. The pyruvate kinase reaction is the last step where the transfer of phosphoryl group from phosphoenol pyruvate to ADP occurs, thereby generating ATP and pyruvate. This is the second substrate level phosphorylation reaction of glycolysis. This enzyme couples the free energy of phosphoenol pyruvate hydrolysis to synthesis of ATP. This enzyme requires Mg+2 and potassium ions. So pyruvate it undergoes tautomerization from enol form to keto form. And if we look at the energetics and products of glycolysis, from one molecule of glucose and 2 molecules of ATP and 2 molecules of NAD plus and 4 molecules of ADP and 4 molecules of inorganic phosphate, 2 molecules of pyruvate, 2 molecules of NADH, 4 molecules of ATP and 2 molecules of ADP and 2 inorganic phosphates are released. So if we look at the net reaction, then there is a net gain of 2 ATP and 2 NADH are formed from the splitting of glucose to 2 molecules of pyruvate. Now this 2 molecules of NADH can be oxidized in the mitochondria under aerobic condition, and the free energy released is enough to synthesize 6 molecules of ATP by oxidative phosphorylation. Under the aerobic condition, pyruvate is catabolized further in mitochondria through pyruvate dehydrogenase and citric acid cycle reactions, where all the carbon atoms are oxidized to carbon dioxide. The free energy released is used in the synthesis of ATP, NADH and FADH2. Whereas under anaerobic conditions, the pyruvate is oxidized, pyruvate is converted to lactic acid in homolactic fermentation or in ethanol to ethanol in alcoholic fermentation. So here the fate of pyruvate is shown. Under anaerobic condition, it can be converted to ethanol or lactic acid, whereas under aerobic condition, it can be converted to acetyl-CoA, which further can be oxidized to carbon dioxide under aerobic condition through TCA cycle. Regulation of glycolysis. Two types of controls for metabolic reactions occur. One is substrate limited and another is enzyme limited. Substrate limited. When concentrations of reactant and products in the cell are near equilibrium, then it is the availability of substrate which decides the rate of reaction. Whereas enzyme limited control involves when the concentration of substrate and products are far away from the equilibrium, then it is the activity of enzyme that decides the rate of reaction. These reactions are the one which control the flux of the overall pathway. There are 3 steps in glycolysis that have enzymes which regulate the flux of glycolysis. These enzymes include hexokinase, phosphofructokinase, and pyruvate kinase. Phosphofructokinase 1, its activity is controlled by a complex allosteric regulation. This reaction commits the cells to channel glucose to glycolysis. ATP is the end product of glycolysis as well as it is the substrate for phosphofructokinase 1. In presence of high concentration of ATP, ATP binds to inhibition site of phosphofructokinase and thereby decreases the activity of enzyme. Whereas AMP, ADP and fructose 2,6-bisphosphate, they act as allosteric activators of this enzyme. Activation of enzyme by AMP overcomes the inhibitory effect of ATP. 2 other enzymatic activities are involved in the regulation of phosphofructokinase. Adenylate kinase, it readily equilibrates 2 ADP molecules to 1 ATP and 1 AMP. Any decrease in ATP and increase in ADP results in an increase in AMP concentration, which activates phosphofructokinase. Another enzyme which regulates phosphofructokinase activity is fructose 1,6-bisphosphatase enzyme, also called as FBPase. It catalyzes conversion of FBP, that is fructose 1,6-bisphosphate, to fructose 6-phosphate, thus reverting back the phosphofructokinase reaction. Here, the high level of ATP can result in allosteric regulation, whereas low levels of ATP can increase the phosphofructokinase activity. So here you can see that ATP citrate, they cause the negative effect or negative control, whereas EMP, ADP and fructose 2,6-bisphosphate, they exert the positive control over phosphofructokinase 1 activity. Substrate cycle or futile cycle. In order to control the flux of glycolysis and to have better regulation, cells have FBPase which keeps degrading the products of phosphofructokinase reaction to its substrate, fructose 6-phosphate. This is called substrate cycle. This is a futile or useless exercise where cells invest an ATP to produce FBP, which is hydrolyzed back to fructose 6-phosphate by fructose 1,6-bisphosphatase. This is a price cells pay to keep glycolysis in check. EMP acts as a potent inhibitor of fructose 1,6-bisphosphatase. Thus, the rate of glycolysis can be increased manifold by EMP as it activates phosphofructokinase and at the same time it inhibits the FBPase activity. Other enzymes, hexokinase, it is allosterically inhibited by its product glucose 6-phosphate. In liver, glucokinase is inhibited by fructose 6-phosphate. Uncomplexed ATP acts as a competitive inhibitor of this enzyme. Pyruvate kinase is the third enzyme which is subjected to regulation. It is allosterically inhibited by ATP. ATP binding to the inhibitor site of pyruvate kinase decreases its ability to bind to phosphoenol pyruvate, which is the substrate for this enzyme. It is also inhibited by acetyl-CoA and long-chain fatty acid. So here you can see that the glucose 6-phosphate is converted into fructose 6-phosphate, and then fructose 6-phosphate is converted into fructose 1,6-bisphosphate by the PFK enzyme. But if the fructose 1,6-bisphosphate is to be regulated, then it is converted back to fructose 6-phosphate by FBPase enzyme. Whereas if the reaction is to be carried forward or the glycolysis is to be continued, then fructose 1,6-bisphosphate is then converted into glyceraldehyde phosphate and dihydroxyacetone phosphate under the high EMP levels, whereas low EMP levels and high ATP will cause the conversion of fructose 1,6-bisphosphate to fructose 6-phosphate. So this is how the levels of EMP and ATP, they regulate the glycolysis. TCA cycle is the another one. TCA cycle, it operates in aerobic condition in aerobic organisms and facultatively anaerobic organisms when oxygen is available. In eukaryotes, it operates in mitochondrial matrix. So here the pyruvate is converted into acetyl-CoA, and acetyl-CoA is then further oxidized completely to carbon dioxide via TCA cycle. Pyruvate is converted into acetyl-CoA by pyruvate dehydrogenase complex, which is a very complex enzyme consisting of three enzymes, enzyme 1, 2, and 3. So here, by the action of pyruvate dehydrogenase complex, pyruvate is converted into acetyl-CoA, and one molecule of NADH per pyruvate is formed. Free energy change of this reaction is minus 33.4 kilojoules per mole. Acetyl-CoA that is formed is then condensed with the oxaloacetate by citrate synthase enzyme, resulting in formation of citrate. This citrate is then subjected to dehydration by aconitase enzyme and rehydration by the same enzyme, resulting in formation of isocitrate. Isocitrate is then oxidatively decarboxylated by isocitrate dehydrogenase enzyme to alpha ketoglutarate. Alpha ketoglutarate is acted upon by alpha ketoglutarate dehydrogenase complex, and oxidative decarboxylation occurs to succinyl-CoA. Succinyl-CoA is then converted into succinate by succinyl-CoA synthetase enzyme, and here the substrate level phosphorylation of ADP to ATP or GDP to GTP occurs at this step. Succinate is then converted to fumarate by succinate dehydrogenase enzyme, and fumarate is then converted into malate by the hydration reaction by fumarase enzyme. Malate is then subjected to oxaloacetate formation by dehydrogenation reaction by malate dehydrogenase enzyme. So oxaloacetate which is formed is then made available for condensation with another molecule of acetyl-CoA. So during this, the two decarboxylation steps occurs, resulting in formation of two carbon dioxide molecules, and oxaloacetate is regenerated. So here, this is how the TCA cycle operates. Aconitase enzyme can be inhibited by fluoroacetate molecule, which is used in controlling the growth of the weeds. Malonate is another inhibitor of the succinate dehydrogenase enzyme complex. So this malonate acts as a competitive inhibitor for this enzyme, succinate dehydrogenase enzyme. Regulation of TCA cycle can be exerted at the level of the pyruvate dehydrogenase complex. So here, the pyruvate dehydrogenase is subjected to inhibition by ATP or acetyl-CoA or NADH or fatty acids, whereas it is positively regulated by AMP, coenzyme A, NAD+, and calcium ions. Citrate synthase is also subjected to regulation. It is inhibited by NADH, succinyl-CoA, and citrate or ATP. Whereas ADP is a positive regulator of citrate synthase activity. Isocitrate dehydrogenase is inhibited by ATP, whereas it is activated by calcium ions and ADP. Alpha ketoglutarate dehydrogenase complex is negatively inhibited. It is negatively regulated by succinyl-CoA or NADH, whereas it is positively regulated by calcium ions. So this is how these enzymes of the TCA cycle, which are the key enzymes, are subjected to regulation. If we look at the overall reaction, the acetyl-CoA is converted into 2 molecules of CO2, and this results in generation of 3 molecules of NADH, 1 molecule of FADH, and 1 GTP. If we look at the summary of ATP synthesis from the oxidation of 1 molecule of glucose through glycolysis, 2 molecules of ATP are consumed and 2 molecules of NADH are formed and 2 molecules of ATP are synthesized through substrate level phosphorylation. Whereas further, at the level of the conversion of phosphoenol pyruvate to pyruvate, again 2 molecules of ATP are formed. So a net gain of 2 molecules of ATP and 2 NADH is there in glycolysis. In mitochondrial reactions, 2 molecules of NADH are formed during the conversion of pyruvate to acetyl-CoA, whereas in the TCA cycle, 6 molecules of NADH are formed. 2 molecules of FADH2 are formed and 2 molecules of ATP are formed, or 2 molecules of GTP are formed through substrate level phosphorylation. So if we look at the overall reaction, the overall gain is 10 NADH, 2 FADH2 and 4 ATP. So from 1 molecule of NADH, 3 molecules of ATP can be synthesized, and from 1 molecule of FADH2, the 2 molecules of ATP can be synthesized. So overall, 36 to 38 ATP molecules can be synthesized from the oxidation of glucose to carbon dioxide completely. So here is the PO ratio. The PO ratio is nothing but the ATP production per oxygen consumed during the glycolysis and TCA cycle. So here you can see that overall 36 to 38 ATP molecules can be gained if the organism grows under aerobic condition in case of microbes, and in case of eukaryotes, the complete oxidation of glucose to CO2 through TCA cycle and glycolysis can result in generation of 36 to 38 ATP. So let us come to the discussion on the questions. The glycolysis and TCA cycles are important pathways to generate energy in the cell. Given below are the statements regarding the production of ATP. Electrons released during the oxidation steps of glycolysis and TCA cycle produce 10 molecules of NADH and 2 molecules of FADH2 per molecule of glucose. This we have seen in the previous table. Electrons released during the oxidative steps of glycolysis and TCA cycle produce 20 molecules of NADH and 4 molecules of FADH2 per molecule of glucose. The coenzymes produced are oxidized by electron transfer chain, and the conversion of ADP and inorganic phosphate to ATP takes place in the intermembrane space of mitochondria. So this is a wrong statement. It occurs in the mitochondrial matrix, and the electrons released during the oxidative steps of glycolysis and TCA cycle produce actually 10 molecules of NADH and 2 molecules of FADH2. So this statement is wrong. So the correct statements are A and C. So if the combination is A and C, that option is the correct one. So answer 4 is the correct one. Next question is, pyruvate dehydrogenase is subject to feedback inhibition by its products in glycolysis. Some of the chemical compounds which might be involved in the process are listed below: NADH, FAD, acetyl-CoA, and acetaldehyde. Which one of the following combinations of above chemical compounds is involved in feedback inhibition of pyruvate dehydrogenase? So if we look at the regulation of the TCA cycle that we discussed, then it is the NADH and acetyl-CoA. These are among the one which inhibits the pyruvate dehydrogenase complex, and others are not involved in the inhibition of pyruvate dehydrogenase complex. So the correct option is A and C. So here also the fourth option, which is having combination of A and C, that is correct. So the correct answer is option number 4. So this was about the glycolysis and TCA cycle. Thank you and happy learning.